Okay, hi everyone. I wish you're all doing great during this pandemic time. Wherever you live in the world, I know that we have people from different time zones. It might be early in the morning or late at night. Thank you for attending this journal club. I'm really grateful for your time. So, Hylan and I are going to represent an article entitled as Contributions of Microbiome and Mechanical Deformation to Intestinal Bacterial Overgrowth and Inflammation in a Human Gut on a Chip that was published in Proceedings of, PNAS, and it was published by Professor Inger's group at 2015. And as you may know, inflammation diseases in intestine is kind of are kind of due to complex interaction of microbiome, whether they are kind of let's say pathogenic microbiome or commensal microbiomes, and intestinal mucus and immune components. So they're kind of component interactions of different components. Complex interaction of different components in the intestine. And so in order to study these interactions and between the, microbiome and epithelial cells, and examine the relative contributions to the potential as the potential influential factors to intestinal inflammation, researchers need a kind of a platform that could mimic, kind of an organ-like platform that could mimic the environment, the microenvironment of the intestine. So this was actually the main focus of this study. So, so uh, they have, this is they have used this microfluidic device to, reach this goal and to, um, see how different uh, types of cells, how different types of components and influential, let's say, factors affect inflammation in intestine. And as you can see that they were able to build this kind of Willy and crypt-like uh, architecture, and that is what we expect to see from in vivo healthy epithelial cells. And as you can see on figure D, there's kind of polarized differentiated epithelial cell that they have made this tight brush border in the picture. But before getting more into the detail of the results they got in this study, I want to go back to their previous study that was published in Lab on a Chip in 2012. They have they have previously showed that this device could culture and could mimic the environment in a good way that could, finally represent and have those epithelial cells quite similar to in vivo epithelial cells that we see in intestine. Uh, their device had three main components. It had two fluidic channels, and they were separated with this porous membrane, and the cells were seeded and cultured over this porous membrane. There were two vacuum chamber that they could induce mechanical strain to this porous membrane. I know that many of you would kind of think that this device is kind of familiar to you uh, because it is the same device that this group has devised and invented in that long on a chip device that is, this device has been used for a long time before this paper, and it's kind of a hallmark or symbol of microfluidic and especially organ on a chip, because and I'm sure that you have seen this video or the following pictures of this device in many of the microfluidic presentations and organ-on-a-chip presentations. So they have used the same long-on-a-chip device with that vacuum chamber in to see whether I mean, culture epithelial cells and kind of use these vacuum chambers that induce this elongation on the cells to kind of recapitulate the environment of the gut. Um, so in terms of manufacturing the device, the upper layer and lower layer of the microfluidic device were similar, and they were prepared by casting PDMS with 15:1 ratio of PDMS to cross-linking on a microfabricated mold that was itself fabricated from SU8 photoresist. Then the porous membrane, this membrane that was prepared by casting PDMS on a microfabricated silicone wafer containing um, circular circular arrays that can produce these arrays in the and make a porous membrane. So they laid this porous membrane over a cured flat selenized PDMS support, and the whole device was cured and was cured for at 60 degrees Celsius for 12 hours. Then the device was exposed to the upper surface of this porous membrane, was exposed to plasma, and the lower surface of this upper microfluidic channel was also exposed to plasma, and these two, these two parts were assembled and bonded with plasma bonding. So then the whole device was incubated at 80 degrees Celsius overnight, and then the support was peeled off, as well as this part of the porous membrane that takes over this surface of this vacuum chamber were torn off, and then again this surface and the upper surface of this lower microfluidic channel were exposed to plasma, and then they were plasma bonded with that plasma, and they were cured at 86 degrees Celsius overnight to produce a bonded and assembled microfluidic gut-on-a-chip device. This microfluidic device were sterilized by flowing 70% ethanol, and it was also and after drying, it was also exposed to UV light and ozone for 13 minutes. Then both sides of this porous membrane was ECM coated by injecting the serum-free DMEM solution that contained type 1, rat type 1 collagen and matrigel. So both surface of this porous membrane was coated with ECM. So this device was capable of producing elongation through, inducing this vacuum pressure. They could produce elongation of by up to 50% strain. And as you can see, when they incorporated different pressures in terms of 2 Pascal, they could have different substrate strain and cell strains. And as you can see, the measured cell strain and substrate strain are quite similar to each other because the cells are attached over the porous membrane, and this porous membrane is kind of moving due to the movement induced by this vacuum chamber. But they have used this 10% cell strain with kind of cyclic stretching in a kind of cyclic stretching form that had 0.15 megahertz of frequency because this is kind of like similar to what happens in vivo for the healthy epithelial cells in intestine. And also you can see here that the epithelial cell, for example, here, exposed to a 30% of the strain. So, and this is the video clip that they had. It shows the strain that is, exposed over the epithelial cell, and you can see this movement of the cells and how they are, tightly integrated to each other. So they have used cockatoo epithelial cells as the cell they cultured in the devices. Um, the culture media was perfused through both microfluidic channel, upper and lower microfluidic channels, in a flow rate that was quite similar to the flow rate in the body, in the intestine, and as flow rate, and as well as the shear stress. And they saw that okay, and then they considered three different groups. One was the static group that used the conventional transwell static device for the culture of the epithelial cells, and these cockatoo cells commonly must be grown and must be cultured in the transfer for almost uh, 3 weeks or 21 days to exhibit differentiated intestinal barrier function. And the other two groups were microfluidic groups. The first one was just a microfluidic device, simple microfluidic device that incorporated only this fluid flow, this media fluid flow that had 30 microliters per hour flow rate. And the third group was exactly that microfluidic device, but it had additional cyclic mechanical strain with that condition that I told you previously, which was 10% of the strain with 0.15 hertz of frequency. And as it is depicted in these phase contrast occluding pictures, you can see that all three groups showed and exhibited, confluent polygonal epithelial cells that were grown in all these three devices. But when we come to this fluorescent microscopic analysis of F-actin distribution and nuclear position, we realize that there is a great difference in this between the groups, because those of the static transwell culture were kind of more flattened and kind of showing squamous in form, but the microfluidic devices showed larger, or let's say taller cell height, and also the cell location was different, the nuclear location was different. And as you can see here in the plot, there is a great difference, a significant difference between the microfluidic, the cell heights in the microfluidic group to the static ones. And it is kind of, let's say, 6-fold increase in the cell height in microfluidic devices. And it gets interesting when we realize that in the literature, the size of this epithelial cells in a healthy, intact intestine in vivo has kind of between 30 to 40 micrometer cell height. So this figure shows that those static conventional transfer culture are not essentially show, represent good, simulation or recapitulation of this epithelial cell in terms of their morphology, at least in terms of their cell height. So this is an important improvement and enhancement that they have discovered in their device. Then, and also there's another important fact here that, this between these two different microfluidic devices and groups, you can see that there isn't any significant increase in the cell height, and this shows that the prominent and important facts that cause this increase in the cell height in comparison to the static group is the shear stress, not this cyclic mechanical strain, because when we add this cyclic mechanical strain, we do not see significant increase in the cell height. So they also tested lower flow rates. For example, they tested up to 10 microliter per hour flow rate, and they saw that in 10 microliter per hour, the cell height is quite similar and quite equal to the static transwell cell culture. And when they increase this 30 microliter per hour up to 100 microliter per hour, they realized that increasing this flow rate more and more doesn't increase this cell height significantly. So this 30 microliter per hour was kind of optimized flow rate for this device, and it was actually in the range of the flow rates that the intestine is experiencing, the epithelial cells are experiencing in the intestine. And then this figure shows exactly the Willi and crypt architecture that we expect to see in vivo, and you can see that this in vitro model, this in vitro device, microfluidic device, recapitulates and simulate and remodel this, let's say, architecture in the in vitro setting. Also, they have measured this TER, which is transepithelial electrical resistance, and this, this TER is kind of a measurement of the integrity of human intestinal epithelial cell. So when it is, if it is higher, then there is a greater integrity between the cells or the tissue that you have built. And as you can see, as the time goes, as the cells are, cultured and grown in these three groups, static, microfluidic, and the microfluidic plus the mechanical strain, in all of these groups, the TER has increased up to a level, and but this level, this maximum amount is higher in the microfluidic groups. But this gets interesting when we see figure 5b, because in figure 5b they have measured the parent paracellular permeability of the intestine epithelial cells using fluorescent dextran FE20. As you can see, there isn't any let's say, significant difference between the first three groups that were static cultured for 5 days, static cultured for 21 days, and microfluidic, simple microfluidic device that cultured for 5 days. And they are all in the same level of, in terms of the permeability. But this gets interesting when you realize that in the literature, in the data published in the literature, is the cockatoo cell monolayers that were cultured in the transwell, in the static groups, do not display, they do display lower paracellular permeability values in comparison to those observed in the human intestinal epithelial cells. So all of these three groups, the static one, the static two, and the microfluidic, simple microfluidic culture are not kind of in good agreements with in vivo, let's say, setting in terms of permeability. But when they have used this, cyclic mechanical strain with that microfluidic that induces shear stress over the cells, they saw a great increasement in the permeability. So this is another important fact here, that this mechanical strain can increase the permeability and could behave better in terms of the, permeability, and they could recapitulate the in vivo microenvironment better. They also measured the specific aminopeptidase activity, and they realized that these microfluidic devices has greater number of cells differentiated, and from cockatoo cells to the epithelial cells. And this is another important fact, that this microfluidic device that incorporates this mechanical strain can induce more differentiation. So this was the history and background of the device, and they've showed that they can build and they can recapitulate this villi crate-like polarized and let's say differentiated epithelial cells that have these tight junctions with each other. So from now on, let's give the stage to Hyun to represent the results.
Can you see my screen?
Yes, yeah, it's fine.
Okay, I will take it from here. So based on the got on the chip model that Alireza had presented, so they were trying to mimic the human intestinal model, and then the first thing they want to research about is the inflammation and the injury on this chip. So they used a non-pathogenic bacteria. Here they use the they use the GFP-EC, meaning the GFP labeled E. coli, and the non-commensal microbe, meaning the bad bacteria, is the EIEC. EIEC here. The EIEC meaning the enteroinvasive E. coli, which this kind of bacteria could cause a very serious diarrhea. And then the third one they use is the LPS, which is an endotoxin isolated from the pathogenic E. coli. So they want to use these three samples to cause the inflammation. The first thing when they inject the bacteria into the upper channel, these bacteria were distributed into the crypt. The crypt meaning between this structure between the villi, but within the 24 hours, these bad bacteria were overgrowth, and they were spread into the apical surface of these villis. So here's a video, and it showed these moving small spots are all bacteria, all bad bacteria. So as the time goes, the bacteria will move from the crypt into the apical surface of this villi. So we can see from this picture that when they add the EIEC into the upper channel, the TER actually decreased very seriously, meaning the bacterial function were destroyed by the EIEC, by the this kind of bacteria. and not to the other two groups, like the good bacteria like GFPEC or the LPS. They don't cause any injury on the villi structure. And from here we can see the morphology that the villi structure in this group, the EIEC group, were actually disappeared in this kind of condition. So another key process happening in the inflammation in human body is the immune cells. So they introduce this PBMC, meaning the peripheral blood mononuclear cells, meaning the immune cells. When they introduce these cells to the upper channel at the same time, they can see not only the bad bacterial group, it caused this severe damage, but also the good bacteria like the GFP E. coli or the LPS, the endotoxin. And the morphology, we can see the structure were also very directly, we can see the structure were totally destroyed. Not only the height of the villi, we can see from here is decreased, but also the lesion area, we can see when they add this PBMC, all the group actually have an increased lesion area compared to the control. So this tells us that both the bacteria, bad bacteria, and the immune cells play a key role in the inflammation and injury process in our body. And then another key hallmark of the inflammation disease is the production and secretion of the inflammatory cytokines. So they tested a group of very common cytokines. In the lumen side, meaning the upper level, these 12 common cytokines didn't have any change. But in the capillary side, meaning the lower channel, adding the PBMC alone could cause the IL-8 had a very significant increase in the capillary side. But when they add the LPS and PBMC together, they can see these 4 cytokines were increased. So what does that mean? When they add these 3 cytokines together or alone, they cannot see any damage to the villi structure. They were basically the same as the control group. But when they add the IL-8, this cytokine into the channel, the villi structure were totally damaged. And when they add the anti-IL-8 antibody to neutralize the function of the IL-8, the villi structure were basically the same as the control group. So this meaning that the IL-8 play a key role in the inflammation happening. Oh, sorry. So after this, we want to know which cells secrete these cytokines, because we know they are epithelial cells and they are PBMC, like the immune cells. So they first run a qPCR sample. The result I didn't show in here. They collect the epithelial cells and the PBMC cell to see their transcriptome level of the inflammatory genes. And the result is that there are 36 inflammatory genes were actually upregulated in the epithelial cells, but there's no change in the PBMC. Well, they also did another marker called TL4. It stands for toll-like receptor 4, which is known to drive the cytokine production. So we can see in they all take picture in this level, in this red line level. We can see the TLR4 is expressed on the surface of the epithelial cells. So combined with the result of qPCR result, we can say that it's actually the epithelial cell that secrete these cytokines. So when they want to move this model into a organ level, there is a very key process in the organ level inflammatory responses is the recruitment of the circulating immune cells, meaning that when the inflammation happens, it will recruit the PBMC from the capillary. So they need another layer of the epithelium to mimic the capillary in the laminar process structure. So they add this monolayer of the endothelium into the upper level to mimic the capillary structure. And they tested this IKM1, meaning the intercellular adhesion molecule 1. It is known to bind the leukocyte or the immune cells when stimulated by the cytokine. So we can see when they add this LPS and then PBMC, we actually can see this IKM1 expressed very highly. So and the quantification, we could see that they were actually increased significantly, not only the IKM1 expression, but also the adhered PBMC, meaning the PBMC were stick into the epithelial cells. So this whole process, meaning the PBMC recruitment, were actually happening. So in other words, the inflammation happened. So when the inflammation is happening, we want to know how to treat this inflammation. So they evaluate the anti-inflammatory using the probiotic bacteria and the antibody. First, when they use this VSL number 3, this is a formula of containing 8 strains of probiotic bacteria, meaning the good bacteria. When you see this magenta 1 here, we can see first, when you add this VSL number 3 alone, we can see the TER actually were increased compared to the control group, meaning the barrier function were actually enhanced by this good bacteria. And when you add this VSL3 and PBMC together, they were very similar to the control group, meaning the PBMC, the immune cells, were actually tolerate this good bacteria, the existence of the good bacteria. So when you see this group, we can see these VSL number 3. Compare this previous figure, we can see that this red one, EIC alone, we can see the good bacteria actually reduced the devastating injury by the EIEC. And when we compare this magenta 1 with this red one here, we can see the injury, although they were a little bit slower than the red one here, but still it can prevent the injury caused by the EIC and the PBMC. So this tells us that maybe the probiotic therapy is only helpful during the early phase of the inflammation, meaning before our body have the immune response. So when you see this morphology change, we can see this two group has no this group basically were same as the control group, but not these the EAC plus the PBMC1. But another therapy is about the antibiotic. So when they add the antibiotic into the upper channel, we can see the height of the villi were actually very similar to the control group. And they were, in other words, they were protected by the antibiotics. Except for the height of the villi, but also the latent area, we can see these two groups. The intestinal barrier function were actually protected by the antibiotics. So the last part is about the bacterial overgrowth. And they see that between these two groups, the one without the cyclic stretching motion and another one with the cyclic stretching, they could see that after like 12 hours, the growth of the bacteria, the green one, were actually like were less than the one without the stretching motion. So this is kind of like the gut disease status, meaning when we have information in our gut, the peristalsis motion might be slowed by the information. So meaning the bacteria were actually might have a chance to overgrowth in the body. So that lead to severe inflammation. So that's the whole biological part of this paper. So I will leave the rest to Alireza.
Yep, thanks. Please, Hyun, do not exit your share screen. You exit.
Oh, okay.
Share it again, and let's go to the next slide, and I will conclude
the
presentation. Thanks. So to put it in a nutshell, they have used a human gut-on-a-chip micro device that was previously designed and showed its potential for culturing epithelial cells with good in vivo-like functionality. Then, which was used in this study to co-culture multiple commensal microbes in contact with living human intestinal epithelial cells. And they use this device to analyze how gut microbiome, inflammatory cells, peristaltic, cyclic, mechanical strain or deformation, these factors, how these factors independently can contribute to intestinal bacterial overgrowth and inflammation. And all in all, this human gut-on-a-chip can be used to analyze contributions of different factors that are available in the microbiome and microenvironment of these epithelial cells in the intestine, and analyze disease mechanisms, and to see which of these factors are doing significant role and a key role in the disease mechanisms. So this was the whole story we got for today. I hope you liked it, and we are available for the questions.